All right, so I'm Mark Rosenzweig. I'm I would say member. I've not been, this is not a group you really want to be a member of, but I've had ALK positive lung cancer since 2013. And I want to welcome everybody. It's a special ALK talk because we're going to introduce you, if you don't know already, Dr. Ken Culver and Professor Mark Muscovich and our own President Kirk Smith. So I'm going to pass it off. No polling questions today, and we're just going to go ahead and get started with Ken.
Now, community, Ken Culver here, your Director of Research. It's a real pleasure to be back. It's been a few months. We wanted to give you an update. Tonight is going to be focused on somewhat of where we are and where we're going with our investments in research and what our goals and are. So be glad to take questions when that question time comes. First, as you may know, last August, Sadia Yaakov joined us as part of our medical team. And she's going to give us a kickoff for our presentation. And I'm just getting it all set here. Okay, there we go. Sadia, all yours.
Perfect. Well, hi, everyone. As Ken mentioned, my name is Sadia. I joined ALK in August of 2024. Prior to joining ALK Positive, I had worked in the industry for over 15 years, worked at CROs, biotechs, other companies primarily focused in oncology trials. My experience has really been in phase 1 and phase 2 studies. And it's been such a joy to join the organization and to work directly with the patients. I think one of the things that I've learned, both during the course of my career and also in school, is that the whole healthcare model has shifted to a patient-centric approach. So truly to understand the patient's needs and to understand some of the barriers that would prevent patients from receiving the best care, what better way than to actually work with the patients. So thrilled to be here today and to be speaking to you guys about the medical committee. And, I think this slide that we have here, I think many people have probably seen this before. We do have 5 medical committees at ALK Positive, and our focus tonight is actually going to be on the Research Acceleration Committee, the RAC, and also the Translational Investigational Group, which is the TIG. Just to kind of recap quickly, our RAC advocates for companies to add ALK non-small cell lung cancer into their development plan, and it also establishes collaborative research programs. I'm also involved with the CTC, and our goal there is to really educate patients about clinical trials and to also increase enrollment rates in our clinical studies. For the TIG, we conduct in silico research and establish collaborations to find new targets on ALK non-small cell lung cancer and therapies for persister cells. And then we also have our ALK Life study that is ongoing, providing real-world evidence on global treatment patterns and the patient treatment journey. And then lastly, the GRC, which is our grant review committee, to contribute to RFP development, grantee selection, and grant oversight. All right. As you all may be aware, we do have a scientific advisory board, which we have formed officially as of the end of last year. And we have 12 members that are part of this board here. We've already had 2 meetings. We had one in Q1 of 2025, and our next meeting will actually be on-site in person at ASCO. And the goal here is to really invest in cutting-edge research, create clinical trial opportunities, and to improve patient care.
Thank you, Sadia. And these are all of these 12 experts are voluntarily participating as a part of our scientific advisory board and have been very generous with their time and their advice. And we're so grateful to all the men and women who were a part of it. All right, now we see So let's talk a little bit more about our medical committees. And we have a number of members who were on, but just to keep it simple, what is the purpose of our medical committees in general is to motivate, facilitate, and enable new therapies. That's basically the core of what our mission is. Yes, biomarkers are important, but basically we, our positive community, don't want to be left out of the really exciting new kinds of therapies that are being developed for lung cancer patients. And I'm going to get into how we're trying to do that. Tonight, the presentation is going to focus on 3 areas. One is going to be about minimal residual disease. And I'm going to go over a little bit about that because we've made our biggest investment to date, the $2 million commitment we made to Breakthrough Cancer is all about this minimal residual disease. I would imagine that's probably almost every patient on this call. We believe minimal residual disease, that there are cells that have somehow been able to stay alive and persist despite the TKI that's in there. And so we're going to come back to that. Then, of course, we're all concerned about TKI therapy resistance that could occur while a person's on any one of the TKIs. And so we're looking, how is that happening and how can that be addressed with new treatments? And then there's immune system. Why does the immune system not kill all the ALK lung cancer cells in your body? Why? Why does that happen? And how can we change that so the immune system becomes the cure instead of having to take drugs? With those as our goals, last year, our medical committees met with 34 different biotechs, 23 universities, and 17 pharmas, and in many cases, multiple times, and in probably about 30% of those, they're ongoing work together. So it's been a very, very busy year, and 2025 is no less as busy as we reach out to companies to have that enable, right, in our mission, enable the development of new therapies in these three groups, in these three areas. So let's talk a little bit about persister cells in MRD. So if you think about an ALK patient who is diagnosed, you go one of three ways, or one of two ways, right? Your TKI doesn't work. We call that primary resistance, and unfortunately that happens even to lorlatinib in the first line. And then there are people who get a response and they come down to this very low level. You may only see a little bit of it on the scans, but that's what we call the minimal residual disease. It's the maximum kill, basically, of the tumor in a patient's body is what we call MRD. But because it's sort of in this really low level and it's hard to detect, scientists and clinicians we don't know a lot about it. We don't know how to quantify it. So if you can't quantify it, then how can you develop a drug there that's going to kill it or eliminate it? So one of the big things about the Breakthrough Cancer Project is one of their initiatives is to understand, define what is minimal residual disease in ALK lung cancer patients. And then, of course, there's this acquired resistance. So patients can be on there for many years. And like some people on this call, you may have multiple of these. You get a resistance and it goes back down to minimal residual disease, and then it goes back up, recurs, and it goes back to minimal residual disease. And one of the things I'm learning about our ALK community is many people have more than one recurrence, and that is a natural part of the history of this cancer. And we want our companies to know that because that drives their business case. So what everybody wants to know on the call is, what is it going to take to get a cure for ALK positive lung cancer? First of all, everybody's got to get diagnosed. And second of all, you have to have access to the TKI treatment. And there's a lot of people who those two things right now, I think since immunotherapy came out for lung cancer, the rate of testing went down. So it's only maybe 60% of 67% of ALK patients are even getting identified. Tragedy. Then you have to have the drug. Then once you're on the drug, what do we need to cure? You got to get rid of those drug-tolerant persister cells. And then you have to have a treatment for patients who don't respond to the TKI initially or have acquired resistance. And then finally, we need a way to activate the immune system to kill residual cancer, which could solve number 3 and number 4. So these are how we're looking at it at the high level. This is as a medical committee. We want to facilitate all these, what it takes to get to a cure. So right now, we are funding 3 different grants to look at identification of how to identify and kill persister cells. With Dr. Carbone at Ohio State, and we already have an ongoing experiments in the lab. They think they have identified one of the reasons why cells can survive with alectinib or lorlatinib. And when we have those animal experiments done, if it's appropriate, we're going to, we ALK Positive plan to invest in the cost to pay to have a clinical trial done. The drug will be provided by a biotech, and we would provide the per patient cost to prove that it works in outpatients, so then the company would take over the funding. We're working with Dr. Polini at Moffitt. They are looking at HER2 is a gene you probably heard of in breast cancer. It's one of the known mechanisms of resistance to ALK positive TKIs. Right now, we're working with pharma to get the donation of a HER2 drug to put with lorlatinib, and Pfizer has been very positive so far. Again, to try to find a way to get rid of those persister cells. And then, as I mentioned, breakthrough cancer, we made a $2 million commitment last year, so $500,000 in 24, 25, 26, and 27. And this is a really, really important project for so many reasons. But let's just start at the top of the slide. This project has more than 20 researchers collaborating to cure ALK positive lung cancer. And one of the things that I liked about breakthrough cancer is they have this right in their slides. Can we drive initial remissions into a durable cure? And I remember so long when I was earlier in my career, we were never allowed as physicians, scientists, or drug developers to use the word cure. And I think so many advances have been made in ALK positive, we believe it's possible. And you can see this is not everybody, but it constitutes really the leadership. And they have 3 aims, as I mentioned, to really define what is MRD. 2 is to confirm it and study it in animals to understand better how we can treat it. And then 3rd, they have created now a national clinical trial framework. So companies who want to develop a drug in ALK can come to Breakthrough Cancer, and Breakthrough Cancer can provide this group of expert sites to facilitate new drug development in ALK positive lung cancer. So this is a breakthrough for ALK cancer. And so maybe we'll ask them to rename their organization. Drug resistance. That is where right now the bulk of our projects are. And ALK Positive here is funding 2, 4, 6 different laboratory projects and 1 clinical trial, which you may already know. Angel Chin is leading the gilteritinib clinical trial at Michigan. It's going to be opening at Cedars-Sinai. It's not quite open yet. And the plan is to also open it in Washington, D.C., and that isn't final yet, so we'll just leave it at that. But you can see the people in the institutions. David Carbone's helping us to study what is on the surface of ALK cells that helps them hide from the immune system or makes them grow despite the TKI. At MIT, we're looking at identifying of antibody therapeutics. Trevor Bavona, who's been a grantee with us before, is continuing his work, looking at what genes are turned on. Jamie Snyder's finishing up metabolic reprogramming, again, looking at how do cells survive the TKIs. We just signed a contract with Dana-Farber and Alice Shaw a couple of weeks ago. It's in her estimation that as many as 30% of ALK patients may develop the loss of this gene called NTAP. And if you lose this particular gene, you become very sensitive to a certain kind of drug called PRMT5 inhibitors. And then we have a gene therapy project, and Jeff Sturm has been working a lot on that. We've been collaborating with the University of Michigan, and we, ALK Positive, have invested some money into those animal experiments to make sure they're done in a timely fashion in the way they and completely. So we're funding all of this. But then our KOLs are also out advocating with companies. So for instance, Christine Lovely was given a grant by ALK Positive a few years ago to study cluster in it. I started off on the wrong one. Nuvectis is a little biotechnology company that's developing an oral inhibitor that targets the YES1 in SARC. That may not mean too much to you as targets, but just know that this drug in the laboratory kills alectinib and lorlatinib resistant cells by blocking bypass pathways that allow the cells to grow. Christine Lovely is collaborating with them, and she's brought the new Vectus group to our organization. And they plan before the end of the year, there is going to be a trial that their trial is going to be amended to allow ALK patients to be able who have developed resistance to alectinib or lorlatinib to join and have the opportunity to get this as combination therapy. So adding this Sark inhibitor onto lorlatinib, for example. And then Ross Camage has been advocating for us, and he was out and he's been talking to Triana Therapeutics. And this is, we don't need to get the details, but they have an ALK molecular glue degrader. The bottom line is it's a way to cause the ALK protein that makes cancer cells grow to cause it to be degraded and destroyed. They're in the animal experiments. If all goes well, thanks to advocacy by Ross, we hope to have that as a clinical trial in 2026. But we're not like every other patient organization. And Mark Muscovitz is going to tell you a little bit more about our own research, and that Mark has been the inspiration and been leading for us to be able to now to identify drug targets in ALK positive cells so that we can go, we ALK positive to companies and continue to push for new therapies for our patient community. Mark? Oh, wait, I was going to do one more before I hand it to Mark. It's a good reminder that lung cancer, when I was in let's start over. When I was in medical school 40 years ago now, oh my gosh, I can't believe I just said that, 40 years ago, we had, when I was taking pathology, the questions on lung cancer were, is it large cell, small cell, or non-small cell? We didn't have any of these drivers. We didn't know them. Then there was a time where you went from thinking of lung cancer as basically one disease with different size cells. Now you had different histological types, squamous and adenocarcinoma. And as you know, at adenocarcinoma, almost all outpatients are adenocarcinoma. And then when you look now, you have all these different kinds of drivers, thanks to the genome project that was launched in 1989 to identify all the genes, is given the tools now. And what this means is that about 70% of adenocarcinomas have a driver gene. Could be EGFR, could be ALK here, about 5%. There's a whole lot of them, and there are companies developing drugs for every one of those. And in fact, in many cases now there are drugs approved on the market. So when you look at this, your first question might be, well, what's the difference? And that is exactly what we are trying to understand, is how is ALK similar, ALK lung cancer similar and different from other lung cancers, so that our patients can get the treatments that are most likely to benefit them. Mark?
Okay, thanks, Ken. So let me spend a few minutes talking about the main project of the Translational Investigations Group. And so what we've begun is a project where we are looking to determine the parts list of ALK-positive cancer cells. And so cancer cells often depend disproportionately on particular proteins in order to survive, to divide, and to evade the immune system. And every cell makes 5,000 give or take proteins or parts that help it grow and survive and for cancer evade the immune system. We can figure out what those parts are by sequencing the RNA molecules in a cell. So DNA codes for RNA, RNA codes for proteins. And if we can decode the RNAs, which we can do with sequencing technologies that have been developed in the last 50 years, we can now infer which proteins are likely to be expressed in the cancer cells. And so what we've done is we've been able to initiate a project that's unique among patient advocacy groups for different types of lung cancer, and I think maybe for all types of cancer, actually. We've been able to do this because of the generosity of donors who have provided funds so we can do the work, because of the generosity and expertise of volunteers on the Translational Investigation Group and the medical committee, and because of the willingness of the board to support this type of research that is usually not pursued by patient advocacy organizations. This slide gives us a listing. I'm not going to read through the list, but it tells us the types of parts we are particularly interested in. So for instance, in ALK positive tumors, we're very interested in whether there are proteins that are turned on in these cancer cells that we could make vaccines against, so that we could provoke the immune system to attack the cancer cells. Are there targets, parts made for which bispecific and trispecific antibodies could be used? Bispecific antibodies have two arms. One of them grabs the cancer cell, and the other one usually grabs an immune cell, bringing it next to the cancer cell and implicitly saying, I think you, immune cell, should destroy this cancer cell. And then the last type of part I'll mention are the antibody drug conjugates. And so what happens there is basically chemotherapy is chemically linked to an antibody, and the antibody binds to basically a protein on the surface of a cell, which you could think of like a grappling hook on the outside of the cell. The antibody binds to it. That antibody and the chemical are taken into the cell, and inside the cell, the chemical is released, and it has a chance to kill the cell. So in order to know whether or not a particular vaccine target or bispecific antibody target or antibody drug conjugate target exists in a cell, we sequence its RNA. And if we go to the next slide, we see the types of information we acquire by doing this. And so each horizontal line here is a different gene, and each vertical zone reflects a set of tumors from a set of particular individuals. The blue vertical lines correspond to ALK positive tumors, the orange to BRAF tumors, the green to EGFR, and the purple to KRAS. So these are different driver genes that are driving the cancer cells to divide abnormally and cause cancer. If we look at the height of these little tiny lines that you can see, the height is reflective of how much of the RNA is made in a given cell in a given tumor from a given patient with a given driver, okay? And what you can see then is if we go down from the top to the bottom, you can see the first 4 lines are 4 genes that are just expressed in ALK positive patients and not in the other patients substantially. The next 4 lines represent genes that are primarily expressed in EGFR patients, but not in ALK patients and not in KRAS. The next 4 lines are genes that are expressed highly in KRAS, and interestingly, they also happen to be expressed in ALK positive patients, but not in EGFR positive patients. And then excitingly, the bottom 4 rows represent genes that are present in all these cancer patients, right? So these are genes that could be targets for ALK positive cancer patients and EGFR positive cancer patients and KRAS and HER2 and BRAF driver patients. So when we look at this, this tells us a number of things. And if we advance to the next box in the animation on the right, so basically what we're doing is we're defining these target genes that are expressed in lung cancer cells. And we're looking at what parts are turned on in those cancers. What we can do with this information then, and interestingly, this research is actually not being done in quite this way by any other groups in the world. This is something ALK Positive is doing distinctively among advocacy groups and distinctively, really, in relation to academia and pharma. And we're doing this because by knowing the parts expressed in the cancer cells, we can pursue item number 2, which is we can then go to companies and say, we know that there's a molecular part in our tumors that you've made a drug against. So we would like you to think about, and we would like to convince you that you should be testing your drug in ALK positive patients because we know the target for your drug is expressed in our cancer. And what we can do as well then is we can use this information to organize clinical trials, and we do that based on not just the driver gene, but also the parts that that driver gene turns on in the cancer cells. So this is really an unusual and distinctive activity of ALK Positive that's been enabled by your support and by the creativity of our volunteers and staff like Ken and Sadia, and by the willingness of the board to support this novel research. And Ken is going to talk about how we then take this information and take this to companies to help lobby them to include ALK patients in tests of their therapies.
Thank you, Mark. So I was able to travel to the American Association of Cancer Research last week in Chicago. And I met with multiple companies. And now you can get a sense of how we're putting this information into practice. So when I would sit down, in this case, it was with the biotech executives. I said, we're using gene expression to advocate for clinical trials. They're right out there. I'm talking to you because I want you, where appropriate, to be testing your drugs in ALK positive patients. So we took the genes that we got off that list that Mark just spoke about, and there were 2, 4, 6, there were 7 genes where they were expressed between 95 and 100% of all these tumors, 20 tumors from ALK and 112 from EGFR. So we could say that, take this example here, B7H3. They're developing a drug against B7H3. I could say to them, based on 132 tumors, every one of those patients is going to be positive. That's 30% of the lung cancer population. So there's a business case made. Then what we do, before we go meet with them, we go on to clinicaltrials.gov. We get all the trials that they have ongoing. So they have 2 different trials ongoing with their B7H3. And then when I met with them, I said, where are you? When are you going to have the information so that we'll be able to know whether your drug works, whether it's safe and would be appropriate for ALK patients? So these first 4 drugs of the 7 they're waiting on FIH, stands for first in human. So it's just being tested in humans for the first time. In most cases, there won't be any ALK lung cancer patients even treated in that phase 1. So we'll be basing, the information off of other patients' cancers. In the case of the HER3 or the ERBB3, right now, there are no trials for us. So in this case, our advocacy is opening a trial that will include patients for ALK. And so we know that ALK patients respond to other companies' HER3 inhibitors, and I don't have it on this slide, but I also had on the version I showed to BioNTech the data from Daiichi Sankyo that shows that 3 out of 3 ALK positive patients responded to HER3 ADC. So, and then finally, they have a bispecific which has 2 different targets on it, as Mark was expressing earlier, and they will be opening a trial. It's not quite open yet, that will allow our patients to go in. And so this is really a bispecific immunotherapy that ALK patients are going to be able to have access to. We're not currently eligible for others. So just as an example, basically, we identify the genes that are expressed in ALK tumors, and then we can go with data to a company to say, here's your rationale for why ALK patients should be included. And in most immunotherapy trials, ALK patients are excluded, and we have to change that. And if we look at ALK and EGFR together, now we have a stronger business case, because all these companies, they want to help patients. I don't have any doubt about that. They have to also get a return on their investment, and they also want to get on the market as soon as possible. So the other thing that we say is the ALK positive patient population is motivated, and if we get behind something, we're going to help you enroll it quickly, because for you, time is money, but for us, time is life. Let's talk a little bit about immunotherapies for a moment, and we'll be done. Right now, we have 2 projects that and they're both with Dr. Chiarli and Dr. Wad. One is to develop a cell therapy treatment. The laboratory experiments are going on. Roberto thinks probably in about a year or so from now, we'll be able to start the clinical trials. Cell therapy is a very complicated development plan. And then of course, there's the vaccine development that we've all heard about. And Mark Awad has been working on trying to develop an ALK vaccine for about 10 years. and they've got fantastic data. I'm going to share a little bit with you. But right now, the issue is getting someone who will manufacture the RNA vaccine. We want to make the vaccine the same way the COVID vaccine was made, using RNA, because it has so many advantages there's lots of different kinds of vaccine strategies out there. So I'm going to come back to that in the next slide. But then there are 3 other companies who are helping us develop trials for ALK. One is BioNTech, and I didn't have it on the other slide because I wanted to discuss it here. They are developing a drug they call BNT116. It's called FixVac. And the data that Mark showed you, we set some of that data to, we showed it to BioNTech and to the investigators, and that helped in a strong way to support. We expect June or July, there'll be this vaccine trial open and for about 30 to 40 patients, trying to use this as a therapeutic vaccine. Then Immunity Bio, this is a connection that we got because Christine Lovely received a grant from us, and she collected the data in her laboratory. The data was positive with a clusterin inhibitor, and Immunity Bio bought that clusterin inhibitor from a small biotech. And so now Immunity Bio wants to do an immunotherapy trial specifically for ALK patients, and that protocol is in development. And we've had multiple discussions with them, and I'm hoping, before the end of the year that will be open. And then finally, I was talking to Christine Lovely, and she said, oh, you really should talk to Regeneron. They've got this really great bispecific antibody that might be good for ALK patients. So we got on the phone. We've had several meetings with Regeneron. We're waiting for a company decision, but I think they really want to add ALK, because right now they're only studying in EGFR. And I guess maybe I'm always saying, what about ALK? But EGFR is great. We're all for helping EGFR patients, but when we've got data, we want to be a part of the drug development. So let me just say a word about the vaccine. I have this title here. So checkpoint inhibitors like pembrolizumab have by themselves show very little activity in ALK positive or EGFR positive tumors. And if you look at the graph, you'll see that the response rate, the ORR means overall response rate. This is from Justin Gaynor's paper. Only 3.6% had a response. And if they had a response, it was really short. And it didn't matter whether the person mattered some if we were a smoker or not, but it was still low. And if you remember these Kaplan-Meier plots, across the x-axis is time. This is time in months. And this is progression-free survival. So how long did the patients go before their tumor started to grow again? And you can see that it was only a matter of weeks. So because there are really 3 studies that all confirmed that EGFR and ALK patients don't respond well to single agent, to individual checkpoint inhibitors like pembrolizumab, nivolumab, and durvalumab, atezolizumab, all those mAbs for these. Our Mark Awad and Roberta Chiarli now started looking at how can we overcome this in animals. And what they did is they took a group of animals and they implanted an ALK positive tumor, and then they gave them no treatment. And again, look, remember, time is on the x-axis. Here it's in days. And this is how long the animals survived. So with no treatment, the animals survived about 25 days. With olatinib alone, because these were intentionally olatinib-resistant cells, with olatinib alone, they only survived about 50 days. And if you added a checkpoint inhibitor, like pembrolizumab, no difference. But once you start adding in the vaccine, you can look here at the red, the olatinib plus the vaccine, now you have about 20% that are still alive at 250 days. And you add lorlatinib plus the vaccine plus a different checkpoint inhibitor, CTLA-4, you cure mice. This was published in a very prestigious journal because they were so rigorously done and important experiments. The problem we've had is we've been ready to start the human clinical trial, but we've had two different biotech companies who promised to make us the vaccine who then changed their mind. So right now we're partnering, this is partnered with Breakthrough Cancer, and their goal is, as soon as we can clarify the funding to manufacture the vaccine, that they're going to do a trial, and they're going to do a trial that's going to be the vaccine by itself, monotherapy. They're going to do a trial with the vaccine plus lorlatinib, which you see here. And then they're going to do a trial with the lorlatinib plus the checkpoint inhibitor plus the vaccine, this group that we see in the graph up here. So that trial is, I'm sure we're going to get the vaccine made this year, sooner rather. Everybody's really trying their hardest, but this is really fantastic data showing that lorlatinib plus a checkpoint inhibitor plus their ALK vaccine has very significant activity and overcomes whatever the problem is with just the checkpoint inhibitor alone. And so we're really excited about it. And I hope by the summit, I'll be able to tell you a lot more about how this has progressed along. So last slide. So from the medical committee's point of view, we're proposing that our investment as an organization and the funding for three things. One, we have to have the laboratory and the clinical data to persuade companies to invest in new therapies for ALK. Two, we're going to need funds to pay for the patient costs for clinical trials. And the third point is those range from about $10,000 to about $50,000 a patient. So if you're going to do a 20 patient trial with $50,000 a piece, it's about $1 million. And after all the changes going on at NIH, as we all know, that have happened since November, I'm expecting those costs probably will go up. So the sooner we can get these trials started, it'll save us on the funding. It's also better for patients. So I'll stop there. Summer and Mark, thank you.
Thank you, Ken. That was amazing. Kirk Smith, our president.
Hey, Summer. Thanks. Well, I didn't know if anybody had. Do we want to answer any questions from anybody? So
let's take it back to Ken, Mark, and Sadia. Ken, do you have any specific? Do you want me to start reading through?
I've got a couple things here. The question about what about lorlatinib? So we'll tell you that with each of the companies we're working with, we're trying to get them to consider all the populations of our patients, those that are on first-line alectinib, those that are on first-line lorlatinib or brigatinib. And but because lorlatinib is becoming the first-line agent, it's always going to be involved. And so we're trying to make sure that our trials don't exclude people who choose Nuvalent instead of lorlatinib and so forth. So that's a work in progress, but just be assured that we're trying to the extent possible to make sure that we can get all the different subgroups of ALK patients involved. But the first trial may not have all of them. We're going to want to get data in one group and then expand from there. Yes, Brian, Anktiva is going to be part of the Immunity Bio sort of cocktail. It's going to be basically with chemotherapy plus 3 immune agents. There's a really good comment in there about quickest profit. At our RAC meeting last week, we had a nice discussion about that, and we are planning to try to work with try to find the right kind of expert in the marketplace who could help us have a more specifically detailed business plan for why including ALK positive is a wise business decision. We all know it's a smart patient decision, and we know it's a good medical decision when we really want to make sure that our case for business is just as strong. And then the NIH cuts are a little hard to know exactly what the full impact is going to be yet. So we'll keep you posted as we learn more, but right now, the obvious decision is going to be that if the indirect reimbursement goes from whatever it 50% to 15%, that 35% reduction is probably going to be put into the direct costs. We as a patient organization, our board has limited the overhead we'll pay to 15%, which is the new one proposed by the administration. So I think it's possible that our costs could go up by about a third. I can't say for sure it'll be different from university to university, but I think it's something that we all should be concerned about. There's one really good question there, of course, about Dr. Lamb's vaccine trial. We are in contact with Vincent all the time. We're following along to see his results. Our priorities are really our trials that have a therapeutic endpoint, so you know if they work or don't work. And in his first cohort of patients, it's a neoprophylaxis, so it's an immune system question that's being asked. So we're all excited to see what that data works with. And also one thing that's changed in this past year is our partner for MRD research is the Breakthrough Cancer Project, of which Vincent is a part of. So I think any decision about how that moves forward will be a collective one, but we certainly are well aware and are keeping in close contact with Vincent about that. The question came up about the gilteritinib study. We have really good news there. Because of the advocacy of ALK Positive, the company that makes gilteritinib is going to actually start a second trial. And many more sites are going to open for gilteritinib, they're hoping by the summer. I don't know where they're going to be yet, but their goal is June or July. So we'll certainly let everybody know. But now that the new Valent cohorts for the second line are closed, gilteritinib becomes even more important. So for patients as an option. And Summer, were there anything else on there you thought were?
All are so great. And thank you everyone for speaking up and asking these questions. I was actually just typing back to someone now that I wanted to speak with. Separately, a patient who he and his wife are both ALK positive, which is mind-blowing. And I'm so sorry to hear that. We're glad to have you in the club, but so sorry that you both are here. Yeah, and I think that is definitely study worthy. Let's talk offline and we can talk further about that and connecting with you with some key opinion leaders. Mark, Sadia, anything? I think you guys and Ken got most of these. These were great. I agree there is a stigma about the name vaccines, but if you go back to Catherine Wu's, this is what the experts are saying. So if we change the name, I don't know that would be beneficial, but that is tricky.
Yeah. And on that point, I am starting to see people change how they characterize their biotech drug. If they were calling it a vaccine, it's starting to morph a little bit, in situ something or, whatever. I think that is becoming more sensitive. And of course, typically in vaccines, we think more about COVID, which is a prevention, just like Vincent is trying to prevent mutations from a certain number of mutations arising in the ALK cancer. But in the case of cancer in general, where we're talking about vaccines, we're talking about therapeutic vaccines. We're actually treating an existing cancer as opposed to try to prevent it. There basically is no, using that definition, there aren't any cancer vaccines approved. And so it's early days. And one of the things that I'm encouraged about is that for the first time that I can recall, the European lung cancer meeting I was at a few weeks ago had a talk on vaccine, but in a 3-day conference, they had one 1-hour talk on cancer vaccines. And most of that was talking about how almost everything has failed. So I think what we're trying to do as a medical committee is think about what are the technologies and what difference, how can they be combined? Because it's not vaccine like we're used to, like getting a measles vaccine or a COVID vaccine. we're asking the vaccine to do a whole lot more than prevent something. And so we're going to solve it as a medical community, but it's going to take us a little while.
That is such a great point and a great question to lead to that. And I think that shows again why these talks are so important, and educating yourself as a patient, as a care partner, so that when you are asked those questions, that you could come back with the word therapeutic vaccine, or that, to explain exactly what it is to people asking. Because we know too, as patients, we get all sorts of unsolicited advice and opinion. Kirk Smith, our president.
I just want to thank Ken again for such a great presentation, and Mark, thanks for your part. It's really interesting to me and I get to hear Ken present a lot of times, and I'm always fascinated. And I hope you notice there's so many comments in this presentation of, well, there's this connection with Dr. Lovely, or there's this connection with Dr. Shaw, or Dr. Camage has suggested this company who has said it wants to talk to us about doing research. I've always found in life in general, life is about relationships, and what we have had the opportunity to benefit from, and Ken was talking about medical school 40 years ago, is that he has a 30-year career in this world, and we would not be able to, not even close, be able to do the things that we have been able to do, and what Ken and the medical committee and what now Sadia are doing without those kind of connections and relationships that he's able to build and reach out to all of these researchers and universities and connections and biotechs without his experience. And I'm going to use a personal one, and I probably shared this with some of y'all before. I was diagnosed in 2013, and at that time, Xalkori was the only drug available. And just after that, I had some issues with Xalkori. I started on a drug called ceritinib, which had been developed at Novartis. Well, Ken was part of the lead team in developing that drug, which kept me alive for 3.5 years before alectinib and lorlatinib and brigatinib were developed. And so, he's not only coming to us with a career in this pharmaceutical development world, drug development world, but it's very specific to what we live with. And so we've been I think we are very fortunate to have him. I also want to give a shout out to Ken on this, because just this winter, he's went to, back in February, he was at TTLC. Then he hopped on a plane to the World Lung in Europe. Then he comes back, and just, was it last week, him and Mark were presenting at a New York City fundraiser. And then he hopped on a plane to go to the AACR conference, which is out in Chicago. And now he's here. He's been working Sunday, because I was emailing him today back and forth, because both of us are in the office working today. And he's emailing me right back because he's working on this presentation for all of us. And then tomorrow he will fly to Seattle to do another fundraiser. So he's flying all the way across the country. So, you will not find somebody that's more dedicated to us and what we are trying to do with this cause than Ken. And now we've also been fortunate enough to have, Mark and Emily Benanzi and Colin Barton and Jeff, who's on here, and a lot of others who are part of our medical committee who helped drive it as well. And now we have Sadia as part of the team. So a big thank you, Ken. Beyond putting together a good presentation, you do a lot of other things great. I told you I was going to say nice things about you tonight.
Those are way too nice. Thank you, Kurt, for that generous. And of course, for me, it's a labor of love and a privilege.
Yeah, well, I'll add too, because I missed saying thanks to, because I'm looking down here at the bottom of my screen and I see Ray Hall, and it's all about relationships. It was Ray Hall's relationship, who is Summer's dad, who had worked with Ken. So, we give shout out to Ray every time to thank him for getting us connected, but it's all about connections in a relationship. So before I hand it off to Katie to go over what we're looking at for ALK Positive Awareness Month in June and what we're planning to do, I wanted to also say that none of the things that Ken has presented to y'all, all these projects that we have been investing in, would be possible at all without all of you guys. and all of you who have donated, who have raised money. Last year we raised $3.5 million. And that was right at $1 million more than we had raised the year before, the most we had ever raised, and we're able to fund these kind of projects and make these kind of things happen. And a lot of that was really kicked off at ALK Positive Awareness Month last year when the Breakthrough Cancer project was kicking off. And so Ken just gave you a little bit of update on that. There will be more of that at the summit. So really, all of you are as much a part of this. I mean, the theme to our summit for the past 4 years has been stronger together, and that could never be more true than all of what you see happening here and with all of you guys.
We will hand it over to the fabulous Katie Delaney, our philanthropy manager, who I am so grateful is on our team.
Thank you, Kirk and Summer and Ken and Sadia and Mark and Jeff and everybody on this call, because each and every single one of you have a part of this story to share, and we're so grateful and excited to present the ALK Positive Awareness Month this June. So everything you just heard is exactly why we're so excited to share this year's ALK Positive Awareness Month campaign and our Powered by Patients campaign. Every dollar raised, every breakthrough made is a story. It's your story. Just like Summer's, how she's powered by time with her family, watching her kids do what they love, and helping others become their own best advocate. This June, we're inviting you to take part in something powerful, whether it's sharing what powers your journey, creating a fundraising page, or helping create awareness. We want to celebrate you in this mission. Our goal is $500,000 to keep powering the research you see here today and to keep pushing it forward. Just like as Kirk mentioned, we are stronger together. So let's see what we can do to help share how we can create and how you can take part in this month's campaign. We have Yvonne, our board member.
My name is Yvonne Diaz. I'm an ALK positive patient diagnosed in August 2021. I'm a member of the ALK positive board, and my background is in communications and PR and social media. So I'll be taking you through how you can use social media to power your campaign, and also just helping us champion advocacy and raise more awareness. You've already been seeing our beautiful cards here where we have a great example from Michelle Hendricks. On the next slide here, so really the aim of social media is really here to help power everything we're doing this month. So for you, if you are fundraising, this is the best way of really sharing your story and creating more awareness and really doing your fundraising. So I encourage you to ask for one of these social media cards. And basically you can see we're using our hashtag powered by patients, which is really very appropriate for this climate, as Katie's been sharing in the earlier briefing. And really, it's here to support everything we're trying to do. We're trying to, at the end of the day, raise more awareness for ALK-positive lung cancer, show all the faces that are impacted by this disease, both patients and our families and friends, and of course, the need for more research. So we're really here to establish a more emotional connection by using that photograph that you select. And this is, again, here to facilitate all of your donations and your fundraising plans. So in terms of how to do this, we're making it really, really simple. We have a Google form, and I hope that Summer or Katie are going to be sharing the link in the chat, and we'll be putting this in the support groups. And we also have an email address where you can contact us with any questions. But in here, you're going to find it your one-stop way of requesting everything. So we're going to ask you for, first of all, how you want your name to appear on the card. So that's one thing, where you're from, and then also picking those 3 phrases here. So in Adam's example here, you see he selected what powers him. So it's his parents, his girlfriend, and also a really specific point here around the will to live. So that's perfect, really. But it can be about anything. So, and then we're going to be looking for that perfect photo that brings that emotional connection to your fundraising. So look out for that link, and please do request them. In terms of how to do it, really, what we find works best is a month-long campaign. It is ALK Positive Awareness Month, so obviously starting on before or just after the first of the month is ideal. If you open with your story, your why, nothing really, touches people more deeply than understanding your personal connection to this disease, and why you want to see more research. Your personal networks are best. So a lot of people do these things on Facebook. Sometimes it's LinkedIn if people want to go to their professional networks, Instagram. And some people just like texting their friends the links or even emailing. So whatever works best for you is the best way of fundraising. And, in terms of what more to do beyond once you share your story, obviously you can celebrate your progress. You can thank your donors, share how well you're doing on your target, and also you can share other people's stories. So if you're following all our accounts on the various social media platforms from Instagram, Facebook, Blue Sky, Twitter, LinkedIn, you can just reshare our content. So if you're thinking, what more can I share? There's nothing more powerful than sharing other people's stories as well. We're also going to be having facts about ALK positive lung cancer, fundraiser stories, and more about what we're hoping from a research point of view. So there will be plenty of content for you to share.
Okay, and here she was going to mention how we would love to celebrate everybody who is fundraising. So that being said, we do have a card that we'd love to highlight everybody who is taking action by hosting a fundraiser, whether that's participating in a race, hosting a trivia night, setting up a golf tournament, or just simply sharing your link and sharing your story. It all matters, and it's all such a great way to share your voice. If you happen to be hosting a fundraiser and I reach out, please don't be scared. I'm only trying to celebrate you. But if you have a fundraiser that you would like to share, please do reach out and share it with me so that we can honor you and celebrate your success on our social media channels. You'll be featured in a card just like this. And if you are tapped out on fundraising, please support awareness by sharing our posts across Instagram, Facebook, Twitter, LinkedIn, and Blue Sky. As you can see, we are using every channel we can to make the greatest impact possible. So we truly appreciate your participation and your support this June.
Thank you so much, Katie and Yvonne. If there are any questions, definitely put them in the chat, and we will address those. And again, this is powered by patients. So whether you're raising awareness, whether you're raising dollars, it's taking back your power, because we all lost that when we got this diagnosis, right? So let's show the world what we're made of. Or whatever you're comfortable with. It might even be just a nice momentum for you if you're not out there on social media. So no pressure any which way, and we are here to help you and celebrate you, just like Katie said. It's like my dogs know the hour is up over here, everyone. So thank you so much to our medical committees. I always tease that it's the while we're sleeping, this is what's happening behind the scenes. And I get so excited, as I'm sure many of you do, as Mark is talking, showing those charts and how you can see drug interactions, and then we go after those companies. And Ken shares with us too about how when they do shoot him down and say, oh, ALK is all set, that he pushes the envelope and says, no, they're not all set. And I think that's important for all of our voices too.
One comment before we leave.
Please.
And that is, Ken, you've been with us for a little bit, and I was wondering your perspective of what it's like to work with patients as you're a forefront to us. Like, what does that give you?
A sense of urgency.
A one-liner. All right, one word.
So, but when I meet with the pharma people, I tell them I work with patients. Weeks matter. I said when we were in pharma, if we said we could do a trial in 2 years, that was fast. I said that's not fast. So I'm trying to bring that sense of urgency in a very real way to people, where their fast is 2 years and our fast is 2 weeks. And of course, the current environment with the uncertainty, what's going on in the FDA and all that, it just makes it all that much more complicated to know where are the roadblocks in bringing the drugs along. And so that's something we're trying to get an assessment on, because everything, compassionate use, everything you do has got to have FDA approval. So it's not, so we're in a vulnerable position right now, and we as a medical committee, to the extent we can lend any strength to moving things along, I know we will.
Thank you. And I'm sure everyone on this call thanks you. And I'll let Summer close us off.
We have a 2-week break. And then we will have 2 pharmacists on, 2 thoracic oncology pharmacists, actually, from the University of Pennsylvania. They actually have helped both Dana Corrido and I. So we're excited to bring them to you to talk about drug interactions, combating side effects, how about using natural homeopathic remedies. So they'll touch on all of that, and that's in 2 weeks, and we are looking forward to that. So bring your questions. They are definitely a vital partner in your care team. So thank you all. Thank you to our medical teams, to our social committees, our marketing committees, our philanthropy manager, Katie, thanks for leading us. And to Kirk, our president, and Nancy, who couldn't be with us. Thank you all so much. And Mark and Dana as well. So thank you all. If you'd like to unmute and give some thanks, we will see you in 2.
Thank you very much.
Thank you.
Thank you.
Bye.
Thank you.
Thank you.
Have a great night, everybody.